Hello, and welcome to the Chapter 12, Parts 1 and 2 online lecture on the cell cycle, cell division, and mitosis. You should use the information in this lecture to complete the Chapter 12, Parts 1 and 2 guided notes, which of course you should complete before you attend class. There are certain times in the life of a cell when it's necessary to increase their numbers or to decrease their numbers. Cells can increase their numbers through a process known as cell division, and you guys have all heard of cell division before. This is where one cell will divide into two or more daughter cells, and they pass on their genetic instructions to that new cell. Single-celled organisms such as paramecia and bacteria use this to increase the number of cells in their populations. Multicellular organisms use cell division as well. This is useful in healing a wound, for instance, or in growing our bodies. Now, believe it or not, sometimes it's important, especially in multicellular organisms, to decrease the number of cells that exist. We refer to this process as apoptosis, or more dramatically, programmed cell death. Sometimes it's also called cell suicide. In this process, special signaling molecules will bind with special receptors on the membrane, and that will cause the cell to basically implode. So the membrane does something called blubbing, where it gets really leaky and lumpy. The cytoskeleton begins to break down inside, so this thing starts to fall in on itself, and the whole cell will fall into these little vesicles filled with different cell components. So the cell just breaks apart. These little components can be recycled into other cells. Now, why in the world would you ever want your cells to kill themselves? That seems really counterproductive. I mean, the more cells, the better, right? Well, not necessarily. Sometimes it's counterproductive to have too many of a certain type of cell. Sometimes you have too many of a certain type of cell in the wrong place at the wrong time, and you need to get rid of them in order for the body to function properly. Let me give you two examples of this. Now, hopefully you guys got to raise or at least see tadpoles when you were a little kid. You know that baby frogs start out looking a lot more like fish than they do frogs, and at a certain point they lose these tails and grow legs. Where do the tails go? They don't just fall off. Well, what's happening is that the cells inside these tails are going through apoptosis. They are breaking down, they are falling in on themselves, and then the body can reabsorb them and use those materials to grow legs. A similar thing happens in human embryos when we are very tiny. We all start out with these hands that look like little paddles. They look like little paws, and there's skin between the fingers. So we have webbed fingers. At a certain point in development, the webbing between the fingers begins to go through apoptosis. Those cells die, and that actually separates the fingers from one another. And yes, it is true that occasionally a baby will be born with webbing between the fingers, and oftentimes the parents choose to have that corrected surgically. Another important reason that a cell might want to go through apoptosis is if it's damaged beyond repair. Consider this. What if a cell or a group of cells is permanently infected by a virus or a bacterium, and that virus or bacterium is just going to spread to other healthy cells? In that case, it's probably more beneficial to kill off those cells, to sacrifice those few cells to save the rest of the organ. And so that's often what the body will do. This also applies to cancer cells. If the cancer is caught at an early enough stage, sometimes they can be signaled to go through apoptosis and break down instead of continuing to divide. Other times, cancerous cells don't receive that signal or ignore that signal and will continue to divide and grow an even bigger tumor. Before we dig into cell division, there are several important ideas that you really need to understand, and the first of these is the cell cycle. The cell cycle represents the life of one cell. So it's all of the events that occur during a cell's lifetime, all of the proteins that are made, all of the processes it participates in, all of the events that occur. Now, measuring the life of a cell can be a little tricky. Different kinds of cells live different amounts of time. So, how do we do it? Well, scientists define the life cycle of a cell, one cell cycle, to be all of the events that occur from the time the cell is divided until it divides again. So, from one division to the next, all the things that the cell goes through. That's the cell cycle. The cell cycle is divided into two major phases, known as interphase and cell division. Cell division, of course, is that period of time when the cell is dividing into two, it's reproducing. At the end of this phase, we say that a new round of the cell cycle has begun. Interphase represents all of the rest of the events of a cell's life, when it's making proteins and doing its job and so on. Now, interphase is another example of something that's poorly named in biology, because the other term for interphase is the resting phase. Why would you call it the resting phase? It's not resting at all. Cells are really, really busy during interphase doing all of their life stuff. Well, there's a historical context for this. When the very first microscopes were developed, they allowed scientists for the first time to actually look at living cells, and that was very exciting. Cells were especially exciting to observe during division because you could see stuff moving around in there. The cell divides in two and changes shape. Really fun to watch. But the rest of the time, when the cell's not dividing, yeah, they're just kind of sitting around not doing anything, according to those early microscopes. So they named it the resting phase. Today, of course, we know that the resting phase is sort of a misnomer, that there is actually a lot of stuff going on inside cells during interphase. But sometimes you still hear that old term being tossed around. Interphase can be divided into three smaller phases. The first of these is known as G1 or gap 1. Now, gap 1 begins at the end of division. So at this point, the cell is going to recover from division. That probably means it's going to need to grow some new organelles that weren't divided up properly, make a lot of new proteins, and kind of increase in size as well, and kind of recover from that process. Now, while the cell is progressing through G1, it's also, of course, going to be doing its job. That can mean different things for different types of cells. Liver cells, for instance, their major job is to help detoxify the blood, whereas a brain cell's major job might be to send communications, to send messages between cells. The second phase of interphase occurs right at the end of gap 1 and is known as the synthesis or S phase. Cells only enter this phase if they intend to divide again, and the reason for this is that it's during the synthesis phase that the cells begin to make their first preparations for division. Synthesis means to build something. So what are they building? Well, during the synthesis phase, a cell is going to copy its DNA. So it's building DNA. This is that process of DNA replication that we talked about back in chapter 16. So in this point, the cell will copy its entire genome and hang on to those extra chromosomes in preparation for eventually dividing and sending one set of that DNA onto one cell and the duplicate set onto another cell. Also during this time, the cell, of course, has to do its job. It can't stop and take a break from its job, even though it's replicating its DNA. I'm sure you remember this, but just to review, this is what DNA replication looks like. I'm sure you remember that it's the enzyme helicase that actually opens up this piece of DNA and makes the replication fork, and that there's a whole host of other enzymes, including DNA polymerases and ligases and others, that actually do the copying. The timing of DNA replication relative to division is also kind of interesting. There's actually a pretty big gap of time between the two processes. Think of it this way. If cell division is scheduled for, let's say, noon on a particular day, DNA replication will happen long before. It will happen at, let's say, midnight the night before. So the cell actually carries double copies of all of its chromosomes for a long time before division occurs. The third phase of interphase is known as G2 or gap 2. This phase is the shortest part of the cell cycle. During G2, the cell is going to make its final preparations for division. So at this point, it's going to build all the machinery that it needs and make sure it has all the proteins and things like centrioles that it needs to actually divide. Now, of course, during this time, the cell is still doing its job. It can't take a break even during G2. But once it finishes G2, it's going to be all ready to divide. The last phase of the cell cycle is known as the M phase. This is the division phase, the time during which a single cell, called a parent cell, is going to divide into two or more daughter cells, depending on the type of division it's undergoing. The cytoplasm and the organelles get split into two or more parts, approximately evenly, although it's not always exactly evenly. And the genetic material is going to be split into even parts and passed on to each of the daughter cells. Cells need to be able to very carefully control how quickly they go through the cell cycle. In other words, they need to be able to carefully control how quickly and how often they divide. If a cell doesn't divide at all or divides very slowly, it can cause a problem with tissue replacement, with healing. But if a cell divides very quickly in an uncontrolled fashion, it can cause things like this: tumor growth, cancer. Cancer really is a disorder of the cell cycle. The cell is dividing too fast, it's not dividing in a controlled way, and that can cause lots and lots of other problems. The rate at which different kinds of cells go through their cell cycles differs depending on the type of cell you're talking about. Skin cells, for instance, divide very rapidly. Here we have the dermis of the skin, one of the deeper layers of the skin. And these cells down here are going to divide, and that's going to push these other layers of skin up. And the layers of skin on the outside, the epidermis, get sloughed off all the time. You lose those all the time and are constantly replaced. For this reason, skin is very susceptible to developing things like cancers. It's dividing all the time, rapidly anyway, so it's really easy for it to get out of control. On the other hand, other kinds of cells hardly divide at all. Cells like nerve cells rarely divide. In fact, some of the nerve cells that you are born with, you will die with. They live as long as you do. Cells use something known as the cell cycle control system to ensure that they are dividing and going through their cell cycle at an appropriate rate. Now, there's not actually a little clock built into a cell, but what cells actually use are groups of molecules that work together and they regulate the speed of the cell cycle. These molecules rise and fall as the cell goes through its different stages, and they can act on the cell in a number of different ways. Some of them act on the cell by sending signals, some of that cell communication we saw in a previous chapter. Others help control gene expression of key genes that allow cells to go from one phase to the next. The cell cycle control system depends on something called checkpoints. Checkpoints are these moments in time when the cell sort of pauses and takes a look at all of its chemical components. And it makes sure that at that time it has all of the chemical things that it needs to proceed into the next phase of the cell cycle. There is a checkpoint at the end of G1. There's a second one at the end of G2, just before the cell begins to divide, and there's even a checkpoint in the middle of mitosis or cell division. And at this point, the cell pauses and makes sure that all of the machinery of division is hooked up correctly and that division is proceeding correctly. There are a number of molecules that work together to make up this chemical clock, including things like protein kinases, cyclins, and some molecules called growth factors. The way this works is that these molecules will rise and fall as the cell goes through its different phases. They work sort of like hormones in a woman's monthly cycle. You know how Hormones go up and down at certain points during the cycle, and that causes different physiological events to happen in the woman's body. Well, on a much smaller scale, we have these chemicals that rise and fall at certain times, and at the checkpoints, the cell will take a moment and look at all the different chemicals. And if all of these chemicals don't have their proper levels at that point in time, it will not proceed into the next phase of the cell cycle. Of the checkpoints, the G1 checkpoint is actually the most important. And that's because this is the point at which the cell decides whether it's going to go on and divide and reproduce or not. The G1 checkpoint occurs at the very end of gap 1, the G1 phase, and it's at this point that the cell looks at its chemical levels and kind of makes a decision as to whether it will go on into S and G2 and divide, or whether it's not going to divide. If it's not going to divide, it will be diverted into another phase of the cell cycle known as G0. This G0 phase really is the G0 phase. It's not the go phase, that's a zero. In fact, it's more like the stop phase, because when a cell enters G0, it's still going to do its job, whatever its job is, but it's not going to worry about division. So it's not going to copy its DNA, it's not going to build centrioles or any of that other stuff that happens in the S and G2 phases. Cells can enter and leave the G0 phase as needed. For instance, cells such as muscle and nerve cells rarely divide, so they can go into G0 and just stay there pretty much indefinitely unless something happens and they need to divide. Other cells will come in and out of G0 fairly frequently. However, let's say that you've been on a really nice diet, you've been eating really nice and healthy, and your liver cells have been in G0 just trucking along. They don't really need to repair themselves very much. But let's say you have a really super fun weekend and you drink a lot of alcohol and eat some bad for you foods, and now your liver has to do an extra amount of work. Some of those chemicals, such as alcohol, can damage your liver cells. And so at that point, the cell needs to regenerate those cells. It needs to come out of G0 and get busy and divide to make more liver cells. So at that point, it can come out of G0, blast through that checkpoint, go through S and G2, and divide some more. When that has kind of stabilized, it's able to go back into G0. So G0 is kind of a holding pattern that allows cells to regulate how quickly, how often they divide.